ATP synthase, enzyme complex for ATP generation. ATP synthase, enzyme complex for ATP generation. Adenosine triphosphate, commonly known as ATP, is the energy currency of life. It is a molecule that powers various cellular processes, enabling organisms to perform essential functions. But have you ever wondered how cells generate ATP? The answer lies in a remarkable enzyme complex called ATP synthase. In this article, we will explore the structure, function, and importance of ATP synthase in the generation of ATP, shedding light on one of the most vital processes in all living organisms. ATP synthase, also known as FOF1 ATPase, is a molecular machine present in the inner mitochondrial membrane of eukaryotic cells and the plasma membrane of prokaryotes. This enzyme complex consists of two main parts, a transmembrane component, Fo, and a soluble catalytic component, F1. Let's delve deeper into each of these components to understand how ATP synthesis is facilitated. The Fo component of ATP synthase forms a proton channel or pore through the membrane. It consists of a rotating ring of C subunits embedded within the membrane, proton binding sites, and a central St. Ulk. This subunit acts as a turbine, capturing the energy from the movement of protons across the inner mitochondrial membrane or the plasma membrane. As protons flow through the Fo component, the C ring rotates, transmitting the energy to the F1 component. The F1 component of ATP synthase, also referred to as the catalytic headpiece, protrudes into the mitochondrial matrix or the cytoplasm. It consists of several subunits, including alpha, beta, gamma, delta, and epsilon. The central stalk, extending from the Fo component, enters the F1 component, connecting the rotor and the catalytic sites. This connection allows the rotation of the C ring to be translated into rotational movement of the central stalk, leading to ATP synthesis. The catalytic beta subunits in the F1 component play a crucial role in ATP synthesis. Each beta subunit harbors three distinct nucleotide binding sites, the loose, L, tight, T, and open, O, conformations. When AP, an inorganic phosphate, pi, bind to the L site, a conformational change occurs within the beta subunits. As a result, AP and pi are brought closer together, facilitating their chemical reaction to form ATP. Once ATP is formed, it is released from the O site of the beta subunits. The rotation of the central stalk, driven by the flow of protons through the Fo component, is vital for the activity of ATP synthase. The central stalk is composed of the gamma and epsilon subunits, with the gamma subunit being the main rotor. As protons enter and exit the Fo component, the C ring rotates, causing conformational changes in the gamma subunit. This rotational movement is then transmitted to the catalytic beta subunits, facilitating ATP synthesis. The epsilon subunit acts as a stator, preventing uncontrolled rotation of the central stalk. ATP synthase is not only responsible for ATP synthesis but also plays a role in ATP hydrolysis. In certain circumstances, the direction of rotation of the C ring can be reversed, allowing ATP hydrolysis to occur. This process is referred to as ATPase activity, and it is important in regulating the levels of ATP within the cell. The generation of ATP by ATP synthase is an adenosine triphosphate generating powerhouse. It is involved in diverse physiological processes, including muscle contraction, nerve impulse transmission, active transport of molecules across membranes, and biosynthesis reactions. Without ATP synthesis, life as we know it would not be possible. ATP synthase, as mentioned earlier, is a crucial enzyme complex involved in the generation of ATP. Its structure and function have been extensively studied, revealing fascinating insights into the Mechanisms underlying ATP synthesis. Scientists have used various techniques, such as X-ray crystallography and cryo-electron microscopy, to obtain detailed images of ATP synthase at different stages of its activity. These studies have provided valuable information about the arrangement of subunits and their interactions, helping us understand the intricate machinery responsible for ATP production. Research on ATP synthase has also led to the discovery of various regulatory factors that modulate its activity. For example, the protein inhibitor if one can bind to ATP synthase under specific conditions. Preventing the hydrolysis of ATP and conserving cellular energy. Other factors, such as the concentration of inorganic phosphate and the availability of substrates, can also influence the active of ATP synthase. Understanding these regulatory mechanisms is crucial for comprehending the complex interplay between energy production and consumption within cells. In addition to its role in producing ATP, ATP synthase has been linked to several human diseases. Mutations in the genes encoding ATP synthase subunits or regulatory factors can lead to mitochondrial disorders, which are characterized by defects in energy production. These disorders can affect various tissues and organs, resulting in a wide range of symptoms and clinical presentations. Understanding the underlying molecular defects in ATP synthase-related disorders holds promise for developing targeted therapies and interventions. Furthermore, ATP synthase inhibitors have shown potential as therapeutic agents in certain conditions. For instance, some antimicrobial compounds can selectively target bacterial ATP synthase, impairing their energy production and inhibiting bacterial growth. This approach offers a unique strategy to combat antibiotic-resistant bacteria and represents an exciting area of research in the field of antimicrobial development. ATP synthase is truly a remarkable enzyme complex that plays a crucial role in the generation of ATP, the energy currency of life. Let's explore these components further and delve into the intricate mechanism of ATP synthesis. The Fo component of ATP synthase forms a proton channel through the membrane. It contains a ring of C subunits, proton binding sites, and a central stalk. As protons flow through the Fo component, EC ring rotates, capturing the energy from the movement of protons across the membrane. This rotation is critical for the functioning of ATP synthase. On the other hand, the F1 component of ATP synthase protrudes into the mitochondrial matrix or the cytoplasm. It consists of multiple subunits, including alpha, beta, gamma, delta, and epsilon. The central stalk connects the Fo and F1 components and enables the rotational movement of the central stalk to be translated into ATP synthesis. The catalytic beta subunits within the F1 component play a pivotal role in the synthesis of ATP. Each beta subunit possesses 3 nucleotide binding sites, the loose, L-tight T, and open, O, conformat. Ions. When AP and inorganic phosphate, Pi, bind to the L site, a conformational change occurs within the beta subunits, bringing AP and Pi closer together. This favorable proximity facilitates their chemical reaction, resulting in the production of ATP. Once ATP is formed, it is released from the O site of the beta subunits. The rotation of the F1 component is initiated by the flow of protons through the Fo component. This rotation is facilitated by the central stalk, which comprises the gamma and epsilon subunits. The gamma subunit A CTS is the main rotor, while the epsilon subunit serves as a stator to prevent uncontrolled rotation. As protons enter and exit the Fo component, the rotation of the C ring produces conformational changes in the gamma subunit, which is then transmitted to the catalytic beta subunits. This transmission leads to the synthesis of ATP. While ATP synthesis is the primary function of ATP synthase, It also plays a role in ATP hydrolysis. The C ring can reverse its rotation, allowing ATP hydrolysis to occur. This activity, known as ATP OSA activity, helps regulate ATP levels within the cell. ATP synthase is involved in numerous physiological processes, including muscle contraction, nerve impulse transmission, active transport of molecules across membranes, and biosynthesis reactions. Without ATP synthesis, these crucial functions would not be possible. In summary, ATP synthase is an extraordinary enzyme complex responsible for the generation of ATP. Through the coordinated action of its Fo and F1 components, ATP synthase converts the energy stored in the electrochemical gradient into the synthesis of ATP. This essential process fuels the diverse functions of cells and organisms. Understanding ATP synthase not only contributes to our knowledge of bioenergetics but also reveals potential therapeutic targets for diseases related to energy metabolism. Extensive research has been conducted to elucidate the structure, function, and regulation of ATP synthase. Techniques such as X-ray crystallography and cryo-electron microscopy have provided detailed images of ATP synthase at different stages of its activity. These studies have enhanced our understanding of the arrangement of subunits and their interaction